All right, do you guys remember the concept of, oh, go ahead, mommy. Okay, so you recall the concept of ploidy, where a typical body cell in a eukaryotic organism like a human being has a diploidy situation or a 2N, right? And that would be the normal condition for every single body cell. And then we have monoploid, and in a monoploid condition, also known as a haploid, this would be the situation for cells that go through meiosis and end up as sperm cells or egg cells, also known as gametes. Okay? However, there's a situation where sometimes you don't have the right number of chromosomes. Don't forget now that if I were to, let me change the thickness to 2. Let's say I had a cell. Shoot. And in that cell was a nucleus. And my chromatin consisted of 46 chromosomes, 46 CHRs. Okay? And that would be exactly the number that you'd want to have because that consists of 23 pairs that you would get from your dad, or 23, I'm sorry, from dad, not pairs. 23 pairs altogether. One of them would be from dad, and the other 23 would be from mom. I'm trying to think if I have a, there we go. All right? However, there are some situations where meiosis does not actually work correctly in the formation of gametes, so that you might end up with an egg that instead of having 23 chromosomes, which is what it would normally be, right? This is normal. Then you would have an aneuploidy situation, so this would be a normal monoploid. Imagine if you had an egg cell that instead had 24 chromosomes. This would be abnormal, obviously, and we'd call this an aneuploidy situation. So this would be an aneuploid. And it might be that it's not just 24, but it might be 22 or perhaps less than that. Typically, if you're missing more than one chromosome, or even if you're missing one chromosome, usually that egg is not a viable egg and fertilization fails. But there are some situations, as we'll talk about, that sneak through. So what would lead to this? What would allow us to have the situation where we have an aneuploid? And that's what I want to discuss right now. Okay. Oh, and by the way, let's just talk about this for a moment. Let's say you had an egg cell that only had 22 chromosomes, and it came together with a sperm that had the right number, 23. Obviously, when you combine these two things, you're going to produce the first body cell, or what's called a zygote. But that would have an issue, right? That first body cell would not have the right number because you'd have 23, let's just make this, you'd have 23 from dad, but you'd only have 22 from mom, and that would only give you 45 chromosomes instead of the diploid situation, which is the 46 chromosomes that we see over here. All right, so let's see where this all goes wrong. I'm going to go ahead and jump to a new page here. I think Cal wants me to go up and play with him, so I'm going to put this on pause, and then I'm going to play for a little bit, and then I'll come down and finish this up. All right, well, he decided that it was okay to wait 15 minutes until Daddy was done doing this screencast. So, imagine this situation. We have a cell, and in this cell, I'm going to make it not a human cell because it frankly would be, it gets to be a huge pain if we do a human cell. So I'm going to do a cell that only has 6 chromosomes. And I'm going to have a situation here where I have mommy chromosomes and daddy chromosomes. So we have one big long mommy one, and then we have a long daddy one. And then we have a slightly shorter daddy one, and a slightly shorter homolog to that one. Ooh, maybe I made that a little bit too long, so I'm going to stretch this guy right there. And then we have a really short version from mommy and a really short version from dad. So we have a total of 6 chromosomes. This is a diploid cell, and there are 3 pairs, okay, or 3 homologs that exist. And in fact, if I just put numbers on these things, you could imagine that this would be number 1. Oh, this is too fat. Hold on, let me erase that. Okay, I'm going to change it to a smaller. Okay, so right here I'd have this as number 1 from mommy, and this would be number 1 from dad. This would be number 2 from dad. This would be number 2 from mom, and this would be number 3 from mom, and number 3 from dad. Okay, so you understand the homologs that exist there. Now I'm going to go through and I'm going to erase each one of those numbers for a moment because they're going to get in the way a little bit of what I plan to do with these things. Now, the first thing we need to make sure we do is to have these things undergo a replication event, right? Have to do replication. So this pink bomb number 1, I'm going to make a copy of it. And I have to make sure that those two are snuggling right next to each other and they're connected by in the centromere region there, right? So let me go ahead and do that for all these guys. This would be after the S phase of the cell cycle. Okay, so here I have a whole bunch of homologous chromosomes in their duplicated state. Now, don't forget that in meiosis, we're going to have a situation where crossing over would occur. I'm not going to show crossing over here because it just complicates things unnecessarily. But what I do want to show is the metaphase alignment in meiosis 1. So let me go ahead and move these guys. Don't forget that these two are going to line up next to each other, the pink and the blue homologs like that. And I'm going to go ahead and put dads over here, like so. And then put moms here. And I'm randomly selecting these things. Frankly, I could have done it in any combination. But imagine, too, that we have here a centriole, and over here we have a centriole. And I send out these spindle fibers, right, which attach like so. And under normal circumstances, these spindle fibers would pull the chromosomes to the opposite poles. So let me just get rid of the spindle fibers for a moment because they're going to get in the way of things. Imagine that what happens here is I grab these dudes. I don't know if I'm going to be able to grab them all. Oh, I didn't. I messed that up. Oh, hold on. Let me get around this and this. Good. Okay. So these get all pulled in this direction, right? And these dudes all get pulled in this direction. And that would be totally normal because later on you could imagine then we'd split the sister chromatids in half and separate them into 4 different cells altogether. So that would be under normal circumstances. Let me just go backwards. And in fact, let's reestablish those spindle fibers again. So I'm going to attach my spindle fibers. But this time I want you to imagine that instead of spindle fibers coming out of this east side here, no spindle fiber goes to that blue dude in the middle, and an extra spindle fiber shoots across and attaches to the kinetochore of that blue one right there, blue number 2. Now, when the things start to pull apart, you can obviously see this is going to create a problem. So let me again get rid of those spindle fibers and show you what's going to happen here. So in this case, I'm just going to put it on pause for a moment and come back in its Julia Child done state. Hold on. Could you have predicted that's what would happen? Because that's exactly what you should have predicted. You can imagine now, here's the problem. If you have a cell that is just now, it will divide. So let's get rid of the, for a moment, let's get rid of this outer area here. Okay, whoops. Oh. Let's do that. Let's do a better job of this, though, this time. There we go. All right, I'm going to draw a new cell membrane around this thing. It could be like this and like this, right? And then you could then imagine I divide this again because that's exactly what happens in meiosis. You have 2 division events. So then I'd have 4 different cells. I'm going to show you what this looks like in just a moment. Okay, there we go. This is what would result. Now, the problem you have here is that the gamete right here, right, that one has an extra chromosome number 2. This also has an extra chromosome number 2, and this is lacking, and this is also lacking. So none of these are going to be gametes that have the correct number of chromosomes. They're all going to be aneuploids as a result of this, right? So all 4 are going to be aneuploids, which will lead to serious issues, right? If you don't have the correct number, you'll find out that leads to things called syndromes. And those syndromes are things that I'm not going to talk about, I think, today. Well, maybe I am, but I don't think I'll have time, so maybe that'll have to be for another screencast. Keep in mind that we had this event where the spindle fibers didn't break apart correctly in the first part, the first meiosis event. And that process, when things don't break apart correctly, has a very specific name. It's called nondisjunction. So nondisjunction leads to, oh, I didn't spell that correctly, leads to an aneuploidy situation. Nondisjunction. Okay? Now, the nondisjunction happened at this first piece. What if nondisjunction happens after that? Let's take a look at that. Okay, so we go back to our original cell situation here. And this time, let's imagine that we have spindle fibers that attach properly and start pulling things apart, and they pull them apart perfectly well. Okay, so I'm going to, again, get rid of these things. And let me just copy this whole cell and paste it right. Oops, I didn't mean that. Let's paste it right here. Okay. So in this situation then, we are going to have the proper, let's see if I can, it's very hard to select things in this screencast. I don't think I can. Let me just move them one at a time. In fact, I'm going to pause it for a moment and try to move things for you. Hold on. Okay, so these things get pulled away, and then they would lead to two cells, so two larger cells, or smaller cells, what am I saying? So it would look like this. Let me pause again and come back. Okay, so it would look something like this. Totally normal. Looks like everything's going well. But imagine now that you have a non-disjunction event that happens here. So let me go ahead and align things and get us moving in the right direction so you can understand what's going to happen. So you'd have this kind of alignment. And then here we have the spindle fibers attaching once again. But this time I want you to not attach a spindle fiber to the sister chromatid associated with the little tiny guy. And here we'll just have it be totally normal. So we'll have spindle fiber attachment, proper junctions that are associated with this. Oh, that one's really bad. Let me clean that dude up. Make it go to the same point. Alright, so now I think you can predict what's going to happen here. Because in this situation, we're going to have the 4 cells that result. Right? And we're going to have no problem with the longest pink ones being in separate cells. No problem with the medium-sized pink ones. But now we're going to have 2 of the little guys in one and not any in the, let's see if this is A, B. The B cell, right here, is not going to have any little ones. And then if we go over to this side, we'll have long, long, medium, medium, and we have no problem with a little short pink, Okay, so if I go ahead and label these things, and this is C and this is D, you can all see that this one is not viable, this one's not viable, but this one's okay, and so is this one. Right? So while you do have aneuploidy in a couple of these, you have 2 that are perfectly fine, whereas in the previous condition, when you had the non-disjunction event happening in meiosis 1, then you had all 4 were aneuploids, as opposed to what you have right now, which is only 2 out of 4 with the aneuploids. Now, the problem with this is that it can be catastrophic. Not only can it lead to the organism not developing properly, but sometimes it'll just wipe out the organism even before it's born. However, we all know, we're all very familiar with some what are called syndromes that are the result of aneuploidy gametes coming together with normal other gametes. And the one that is most familiar to us is one called Down syndrome. Okay? We'll talk more about Down syndrome in tomorrow's screencast, but for now I'm going to stop right here and I pause it right here. Bye.